Hey everyone! Sorry, I'm really sick today, so I know you're sad that I'm not there, but I am going to still go over the notes real fast. You have a new note sheet at the front of the room that you should have by now. Remember, as usual, note taking skills. Do not write nothing. Draw a picture, underline, highlight, whatever makes it better for you and what will help you remember. Okay, so here's my meme from Valentine's Day. I want to make sure you guys didn't miss out on it. Billy the bacterium had to quickly consider the etiquette for declining a bacteriophage's Valentine. This should make sense now, because we all know bacteriophages are viruses, and they like to infect bacteria cells. So no, the bacteria would not like the bacteriophage's rose DNA. Okay, moving on. So today we're going to talk about mutations, because we finished protein synthesis, right? So you should know what happens when we add nucleotides to help us go from DNA to mRNA to protein. So sometimes we have mistakes that happen, yeah? We call those mutations, and you know that. So we're going to talk about the different types of mutations and how they impact protein structure today. So let's do that. Today we're going to finish up the difference between substitution mutations. There's many kinds we'll talk about, so you should be able to know how to distinguish between all of them. We're going to talk about genotypes versus phenotypes. We're going to talk about this in genetics some more, but I figured I'd just introduce it today because you're going to see these terms thrown around. Also, you should know the difference between insertion and deletion mutations. We will talk about chromosomal rearrangements next class. Okay, here's my other meme. And when he looked at his strand the next morning, one of the bases was gone. Sharing mutation stories. Oh, look at their little eyeballs. Aren't they so cute? Okay. So, mutations. Mutations can affect protein structure and function. Remember, I've already said this a million times, right? A protein's structure determines its function and its job. The minute it is not shaped the same way or incorrectly, that protein function is in jeopardy. Therefore, you may have a non-functional protein. That's why we talk about mutations. Mutations, though, are responsible for gene diversity. So we get diversity through mutations, and sometimes that's a good thing, especially when it comes to evolution and having mutations build up over time. Sometimes those are good things. They cause good changes, and then sometimes they are bad. But this is how we get a source of new genes, so it could be good. There are large-scale and small-scale mutations. So when I say large-scale mutations, that means chromosomal rearrangements. Chromosomal rearrangements means part of the chromosomes can completely be swapped, moved around, or changed. You know now, because I've mentioned this before, chromosomes have hundreds of genes on them. If you accidentally swap one part of a chromosome, you just swapped hundreds of genes, and this could result in genetic loss of information. That's not good. So those are large-scale mutations because they impact many genes. Small-scale mutations are going to impact a single gene most times. And this is what we call point mutations, substitutions, silent mutations, missense mutations, nonsense mutations. Yeah, there's all kinds. So we're going to talk about the different kinds. But usually, small-scale mutations are individual or a few nucleotides get disrupted at one time. Okay, so let's start with point mutations. Point mutations are kind of like one big heading above, okay? So point mutations, this is when we change a single nucleotide. And when we do this, it could also disrupt the nucleotide pair. So bases are complementary. So if we mess up one side, we mess up the other side because DNA polymerase likes to add the correct nucleotide. So that will be a point mutation. If it occurs in a germ cell, that gives rise to gametes. It can be passed down to future offspring. So this is how it could be passed on through genetics. Germ cells. When I say germ cell, that means any cell that is used for reproduction. So that's a sperm cell or an egg cell. So in the point mutation, if it impacts the phenotype of a person, it's going to result in a genetic disorder. We're going to talk about phenotype versus genotype in a second. There are different types of point mutations. That's my cat. Okay, so substitutions, insertions, and deletions. So we're going to talk about these three types of point mutations. Okay, so phenotype versus genotype. In phenotype, when I say phenotype, I am talking about what do you see? The observable characteristics. What are you physically seeing? Are you seeing a white flower, a yellow flower, a pink flower? Are you seeing a black mouse, a white mouse? Are you seeing Drosophila eyes, Drosophila flies? Are you looking at red eyes? Are you looking at black eyes? What are the color of their eyes? These are all things that you can physically see. Now, the phenotype is a result from the genotype, right? So a genotype, it has the word gene in it. This is what you're going to observe in a molecular genetic background, okay? What is the chromosome saying? What is the DNA saying? Those instructions give you your phenotype, right? But they're separated because if I say genotype, that's the genetic code. If I say phenotype, that's what you physically see. Phenotype starts with a P, so does physical characteristic. So think of it that way. All right, so let's talk about substitutions. So what do you think is going to happen in a substitution? It says it. It's going to substitute a different nucleotide. We call this nucleotide pair substitution. So you're going to replace one of the nucleotides and its corresponding complementary partner with another pair of nucleotides, not original nucleotides. Some substitutions have no effect on the protein. They're called silent, and we're going to talk about those in a second. There are also missense mutations where we change amino acids, or nonsense mutations where we make a stop codon prematurely. So we're going to talk about these. Okay, so in silent mutations, this is a type of substitution. There's going to be a change in a nucleotide. So in this example on the left side is no mutation. It's the normal. You should have a lysine. But in a silent mutation, you're going to change one nucleotide. So you're going to change that C to a T. And when you get to mRNA, you get AAA instead of AAG. The thing about silent mutations is, hey, yeah, we had a change. However, you still get lysine. So you have the same exact amino acid. Remember when we talked about redundancy and how there's more than one way to get an amino acid, even if there's a slight change in the codon. That's why there are 64 codons and only 20 amino acids, so that we have some form of redundancy. So this is a silent mutation. It doesn't change the amino acid, therefore the protein is exactly the same. So this means there's no observable effect on the phenotype. So this is known as a silent mutation. They're silent. You don't really see them unless you look at the genetic level. Okay. Here's an example. So the top picture in all of these pictures is going to be a wild type normal example. The one on the bottom will be where we change something. So if you look at the bottom one, I'm going to replace this A with a G. Replace the G with an A. And when I do that, we're going to get a U in mRNA transcription, but we still get a glycine. So we get a glycine in the top and a glycine in the bottom. Sweet. The protein's exactly the same. This is a silent mutation. Okay, missense mutations. Missense mutations are substitutions that are going to change one amino acid to a different amino acid. There are two types of missense mutations. I won't ask you the difference between the two, but you should know what a missense mutation is overall. In a missense mutation, again, you're going to change one of the amino acids because of a mutation. So the missense are on the far right of this graph here. Conservative missense mutations are going to be a point mutation. We're going to change the middle letter from a T to a C. And normally we'd have lysine, right? Over here, instead, we have arginine. We've completely changed the amino acid. In this case, if you look at the structure of this amino acid, it is almost the same as a lysine structure, just slightly different. That is close enough that the chemical structures are so similar, it may not really change the amino acid structure that much. So it's a little bit less detrimental. But if you look at the non-conservative region, look at this one. Look how its shape is more like flat and pancake-like. That is a completely different shape. That gives us threonine instead of arginine. We change the amino acid. In this case, this amino acid will drastically change the shape of the protein because of its chemical elements attached to it. So, missense mutations, you can have two kinds, but as long as you remember, hey, we're going to change an amino acid in this type of mutation. All right, so here's an example. Top one is your normal. We should have glycine at the end here before our stop codon. But in this case, we're going to replace T instead of C. And what happens is we get a serine instead of a glycine. So we've just completely changed the amino acid. Okay, nonsense mutations. This is my favorite because you get nonsense. I love the word nonsense. So in a nonsense mutation, a point mutation occurs where you're going to get a premature stop codon. When you get this stop codon and it happens early, what happens? The protein stops being translated early. And if the protein stops being translated early, you do not have your full functional protein. You have half of a protein, for example. This is non-functional now. So this is a dramatic problem. This type of mutation is detrimental to your protein. All right. So normally, your polypeptide will be shorter than the normal gene and can lead to non-functional proteins. All right. So those are nonsense mutations. Here's an example. So remember the top one is regular normal DNA. Now, if I have an A here instead of a T on the bottom, see the yellow letters? We get a U here in our mRNA. Guess what? That's a stop codon. So we get methionine, the start codon, but we do not get all the following proteins. So therefore, this is a nonsense mutation because the stop codon prohibited the translation of the rest of this protein. Okay, so I'm going to pause a second. I'm going to give you some time. I want you on a piece of paper to describe the differences between all the substitution mutations we just talked about. Remember, there are 3. Don't just tell me the 3. Tell me why they're different from each other. You're going to put that on a piece of paper, and then you're going to turn that paper in for me because we don't have Pear Deck today. So I'm just going to hang on for a few minutes while you do that. Okay, I'm going to give you one more minute. Okay, so I'm going to look over your Pear Deck questions to see how you did on this part, but I'm not going to really go over them this time. I will go over them next time I see you, okay? All right. So let's talk about insertions and deletions. So guess what happens here? You're going to insert a nucleotide, or you're going to delete one. So the addition or loss of a nucleotide, these are insertion and deletion mutations. These mutations end up being disastrous for your protein. They completely destroy everything because you get a frame shift mutation. So if you alter the reading frame, and when I say reading frame, that means you start on one end and you count by 3s. Remember, we have every codon equals 3. If I were to remove part of one of those codons, it would either shift to the left or it would shift to the right, depending on if you subtract one or you add one. You can also add or subtract more than one. You could do multiples of 3 that get inserted or deleted, or 2. It all depends. But this results in an improper lineup of your codons, and that results in every single amino acid down the line being the wrong one. This is why it's disastrous for your protein. One small change shifts everything. Okay, so here's what they look like. Here's an example at the beginning at the top. If we look at the bottom, let's put in an extra A. So let's add an extra nucleotide right here after methionine. Normally we'd have a lysine. In this case, we have a stop codon, O. So in this case, we've stopped the translation protein altogether. That's not good. How about a different example? Let's remove a nucleotide. So let's say I remove an A here. Okay. So if I remove an A, everything gets shifted backwards by 1. Meaning instead of phenylalanine and glycine, we're going to get leucine and alanine now. That changes the whole structure of the protein, guys. So this is why it's disastrous for a protein to have a frame shift mutation. So because I'm not here today, we don't get to do this activity, but next class, we are going to use software to find a human gene of your interest. You get to pick any gene you want. We're going to find the DNA sequence for it. We're going to use some software that we can allow mutations. So I'm going to make mutations in a gene to see how that affects the translation of the protein. So we're going to do that next class because we didn't have time to do it today. All right. So I just want to stress that the phenotype of an organism can be affected by small-scale changes. You just saw a frame shift. One change in a nucleotide can affect the whole sequence of a protein. So just because it's a small-scale mutation doesn't mean it doesn't have an impact. It does. And if you think about this evolutionarily, over time, if mutations keep building up over time, this could definitely alter the genetic code of an organism over time. And that's why, okay, remember, it takes millions of years. It doesn't happen overnight. But that's how we start to get the evolution of new species from different ones. All right. So I want to talk about 2 examples that happen from mutations, a few genetic disorders. Number 1, sickle cell. We've talked about this before, but I never told you the exact mutation. So normal wild type beta globin in a red blood cell in a human gives us a red color, but also gives us the round shape of a blood cell. The wild type beta globin DNA is displayed above. When you have sickle cell, you get a sickle cell shape in your blood cells. See the sickle shape up above? This is a problem. This is due to a mutant beta globin DNA source. So instead of CTC, we have CAC. That is a single point mutation. And it will change your mRNA to GUG instead of GAG. So we get valine instead of glucine. So that's a problem. This gives the cell its sickle shape. And this is extremely painful for patients because these little blood cells that are sickle shaped do not flow through veins properly. They clot, and people get blood clots, and that's extremely painful, but also dangerous. You could possibly die from blood clots depending on where they are. A lot of these people have to have blood transfusions because the sickle cells just keep building up. We don't necessarily have a cure for this. I think there's some medications that can help, but this is one of those disorders that we are trying to use gene therapy for, where we can cut out this mutant part and hopefully put the right gene back in so that it can no longer make sickle cells. I think they tried this successfully in mice, but we're not even close to human trials yet. So it's on the works. It's one goal we had. Okay, so here's the SEM image. You guys know what SEM is. You can see how longly, weirdly shaped these red blood cells are, and those could get really sticky and pack in tight in veins, and they don't float properly. So this is a problem. Okay, the next one I want to mention is cystic fibrosis. So cystic fibrosis is due to a mutation in the CFTR gene. This is a 3 nucleotide deletion, and it results in a frame shift mutation. So we just talked about frame shifts. If you shift all the frames over, it's a problem for the protein. So mutations in this gene are going to disrupt chloride channels that are found in cells. And this is going to keep them from regulating the flow of chloride ions and water across cell membranes. So this really impacts the cells that line passageways of the lungs and the pancreas and other organs that produce mucus. So that this way the mucus becomes really thick and sticky, really thick. And when this happens, it blocks airways, so it's hard to breathe. It can also cause problems for your bile ducts. So I've heard that sometimes when people have cystic fibrosis, they have these weird shaking vests that like shake you up so they can try to get the mucus dislodged so you can get it out better. All right, so this is another type of mutation. Okay, so next time we'll talk about large-scale mutations. We're not going to do that today. Okay, so before I leave you, just one quick thing. Please check the announcement in Canvas because I have told you when your quiz is next week. It is on paper. It is 20 points, 16 questions, 15 multiple choice, a couple fill in the blank, and then you're going to have to do for me a transcription and translation of a specific gene. Not a whole lot, just part of the gene. So I know that you know how to use a codon chart. All right. The study guide's also posted. It's in modules under this unit. It's also in the announcement. Cool. All right. So be good. I will see you when I see you. Goodbye.